1. Master regulators of the body. Architectural blueprint. Hox genes act as master regulators that determine the identity of body segments along the head-to-tail axis during early development. Homeobox domain. These genes contain a specific 180 base pair DNA sequence known as the homeobox, which encodes a protein domain that binds to DNA. Transcription factors. They function as transcription factors, meaning they turn other genes on or off to orchestrate complex morphological changes. 2. The 4 human clusters. Genomic organization. In humans, the 39 Hox genes are organized into 4 distinct clusters located on different chromosomes. Cluster labels. These clusters are designated as HOXA, HOXB, HOXC, and HOXD, which arose through ancestral genome duplications. Commonality. This 4-cluster arrangement is a shared characteristic across most jawed vertebrates, providing redundancy and specialized function. 3. Spatial and temporal collinearity. Orderly arrangement. The physical order of genes on the chromosome corresponds directly to the order of body parts they define from head to tail. Temporal sequence. Genes at one end of the cluster are activated earlier in development than those at the opposite end. Precision control. This spatio-temporal collinearity ensures that the neck, thorax, and abdomen develop in the correct sequence and location. 4. Beyond the AP-axis. Multidimensional roles. While famous for head-to-tail patterning, Hox genes are repurposed for developing other complex structures. Limb development. Specific subsets of Hox genes are critical for organizing the proximal-to-distal growth of arms and legs. Organ positioning. They provide the necessary positional information for the internal placement and shaping of organs, like the lungs and kidneys. 5. Patterning the limbs. Appendage identity. Hox genes of the A and D clusters are specifically assigned to define where the humerus ends and the fingers begin. Numerical code. Higher numbered Hox genes, such as HOX11 and HOX13, are restricted to the most distal parts of the limb, like the wrist and digits. Evolutionary shift. The evolution of the Hox clock allowed for the transition from fish fins to the complex tetrapod limbs seen in mammals. 6. Evolutionary conservation. Deep time roots Hox genes are found in almost all animals, from simple jellyfish to complex mammals, indicating they evolved over 500 million years ago. The bilaterian ancestor. The last common ancestor of all bilateral animals likely already possessed a primitive cluster of these genes. Genetic tinkering. Evolution proceeds not by creating new genes but by changing how and when these ancient Hox genes are expressed. 7. Whole genome duplication. Expansion of complexity. Early vertebrates underwent 2 rounds of whole genome duplication, resulting in the 4 clusters we see today. Functional redundancy. Having multiple clusters allows one gene to mutate and take on a new role while the others maintain the original vital function. Diversity of life. This expansion of the Hox toolkit is largely responsible for the incredible structural diversity found among vertebrate species. 8. Expression, timing myths, embryonic start. It was long believed that Hox genes only function during the brief window of embryonic development to set the body plan. Lifelong activity. Recent research has proven that Hox genes remain active throughout an organism's entire life. Postnatal regulation. In adults, they continue to provide topographic memory to cells, telling them where they are located in the body. 9. Roles in the adult body. Tissue maintenance. Adult stem cells use Hox expression to maintain the specific identity of the tissues they reside in, such as skin or bone. Cellular memory. A fibroblast in the foot remembers it as a foot cell because it expresses a different set of Hox genes than a cell in the scalp. Metabolic regulation. Emerging evidence suggests these genes also influence local metabolism and blood vessel growth in specific body regions. 10. Regeneration and repair. Healing blueprint. When a bone is fractured, Hox genes are reactivated to guide the repair process and ensure the bone heals in its original shape. Wound response. The body uses the embryonic Hox program to recruit the right types of cells to a site of injury for effective regeneration. Medical potential. Understanding how to manipulate adult Hox expression could lead to breakthroughs in regenerative medicine and organ engineering. 11. Summary of Hox significance. Universal language. Hox genes represent a universal genetic language for animal form, shared across the vast majority of the animal kingdom. Clinical relevance. Mutations in these genes lead to severe congenital disabilities, but their study also offers paths to healing. Ongoing discovery. We are only beginning to understand how these genetic architects continue to build and maintain us every day.